Hello, everyone. So I'm Nicolas Lobato-Dozier, and I'm just starting as an associate professor here at Sorbonne University in biophysics. So if you're a fan of physics of life, this talk is for you. If you're not yet a fan of physics of life, it's my job to get you interested about the topic. So this is a work I did in a postdoc between France and Japan, and we're going to talk about neural coding of temperature with a DNA-based spiking chemical neuron. The basic idea is how can we mimic the way biological neurons sense temperature with DNA? And the first two questions before starting that you might have is, how can we actually compute using DNA? And even if we can do so, why do we want to do so? So on the first question, what we use is the fact that DNA is replicable, so this makes it programmable, because you can have production rules that will make a molecular program. So your program, for instance here, will take X as an input and create Y as an output. And this can be done in DNA. For instance, your program will be this type of DNA strands. That is, the first half will be complementary to your input, and the second one will be complementary to your output. And in the presence of your input, you will produce the output. How does this happen with biochemistry? Like this. So what happens is your input binds here to the first part, and then you have an enzyme, a polymerase, that will extend X according to the complementary of this sequence, that happens to be Y. And then another enzyme will cut here, and you recover your input X, and you just created a new strand Y, that is your output. And of course, because you're free on most of the nucleotides here, you can do this for many different X's and many different Y's. So that's for the how. Now, for the why, the easiest answer for me is we need DNA computing when data is DNA. So the first one in the field is, of course, diagnostics, because DNA is a part of us. So there's a lot of people interested in circulating DNA, interaction with microRNA that are cancer biomarkers. But today I'm going to talk, and I think most of people are interested in DNA data storage. So we've had a lot of talks about how you read DNA data, so with different sequencing methods, how you write this data. And today I'm going to talk about how could we process. And what I want to, when I say process, I would like to process in moleculo, because if the point would be not to have to sequence every time you want to make a calculation. And so on that, one of the things you might be interested to do on a database is to do some search and classification. And these are things for which neural networks are great. And the question we might have is, can we have these neural networks be done in DNA? And actually, the field of DNA neural networks already exists since a few years, and there's quite a great work. I think in the next talk, Yannick Rondelez will get a bit more detail on that. But the community has achieved linear classification, more recently non-linear classification. But if you look at these results and you compare to biology, these are not very dynamic. Most of the readouts at the end, it's just an endpoint measurement. And compared to, for instance, sensory neurons, you don't have real transduction. What you're measuring, your input is quite often either a sequence or a concentration, and that's not the case for life. If you look at what happens in life, like temporal dynamics, they're really omnipresent and crucial in neurons. If you look at a single neuron, spikes are very rich in information, because you can get information from the frequency of the spikes, from the amplitude, from the phase, and that's for one neuron. And of course, they work in a network. And if you have a network and you think about activation, inhibition, it's all related to synchronization of such spikes. So that's one of the things we would like to mimic. And the other idea is, how do these neurons actually transduce information from the physical world into electrical signals? And so the inspiration from this project comes from the Nobel Prize of 2021 in physiology of medicine. And they showed that thermosensation was happening at a molecular level. If you look at a biological thermosensory neuron, yeah, I'm going to try to look. Yes, here. For instance, here, the one that is responsible for you feeling when something is very cold and very quickly removing your hand, they showed that it was related to the presence of an ionic channel, TRPM8, that was very sensitive to temperature. And this channel, when it's hot, it's closed, but when it's cold, below a certain threshold, it opens and it lets ions flow. And that's how then it's transformed into an electrical signal. And the idea for this project is, this is happening at the molecular level, how can we mimic a bit of that with DNA? And so we made a chemical thermosensory neuron, where we're using the fact that when it's hot, you have your double DNA helix will break into like single-strand DNA, and also you will boost thermophilic enzymes, so their activity will be higher. On the other hand, when it's cold, you will have DNA that is going to be nicely hybridized as a double helix, and you will have the activity of some enzyme that will be slower. So the outline for the rest of the talk, I'm just first going to introduce you the spike generator, so what's the molecular circuit we use. Then I'm going to show you with some mathematical toy models how biological neuron and or DNA neuron have very similar bifurcations, and I will show you what is a bifurcation. And then we'll see how we can use that to encode information either in a digital way or through the firing rate of these neurons. So the spiking chemical neuron we use is based on a predator-prey oscillator. These are 3 DNA strands. The first one is going to be the template. It's the grass. This is the equivalent of the program that I showed you first. Then you have a strand that is going to be the rabbit, and another one that's going to be the fox. And the way these interact, you first have pre-replication. So the rabbit binds to the grass. It is extended. And so it is extended here, then cut, and you get 2 rabbits. And this can be followed via fluorescence here. So that's for the pre-replication. And then a prey can also, so let's see, yeah, thank you so much. Yes, and this I get, no, okay. So then for the so that's for the pre-replication. Then for predation, what you have is that your DNA strand will be captured, so the rabbit will be captured by a fox. It's extended, but then it's not cut, and the sequence happened to be made so that an extended rabbit is a fox. And so this is exactly like 1 fox eats a rabbit, and you get 2 foxes. And then all these DNA strands can be degraded, and so this is like natural death via an exonuclease. And when you put all these with the right enzymes into a test tube, you get nice oscillations. And what I studied is what happens if you vary the temperature. And the 2 things that we observe and that we're going to try to understand together is how can we understand that the frequency changes and that you have this bifurcation at high temperature here where the system stops. And we're going to see if we just broke the system or if it's normal that it doesn't oscillate. So what we did to understand, we dissected our system into different blocks, and we studied separately the effect of temperature on each one of these blocks and inputted that into the equation. So this is just the predator-prey equations from Locta-Volterra. And we extracted experimentally these coefficients that are the variation with temperature of the pre-replication, the predation that acts as an inhibition, and here the degradation. And for the predator, it's produced by the predation and is degraded. And these equations, they're not trivial, and it's not so easy. So the first thing when you have these kind of differential equations, the first thing you try to find is understand and study the equilibrium points. So these equilibrium points, we find them at null-Klein's intersection, and I'm going to explain that. So to study this kind of system, you can make what is a phase space. So on one axis, you're going to have the concentration of prey, on the other one, the concentration of predator, and on each point of this graph, the arrow is going to be the derivative of both these, is going to be the derivative of the prey and the predator. So basically, it's going to give you the trajectory in this plane of your system. And then if I take just the first equation being 0, is going to be my green line. The second equation is going to be my blue line. And to study an equilibrium point, I want both my equations to be 0. So it's going to be the intersection of both my systems. And if you look at the trajectories, all the arrows, they seem to do funny stuff around this point. So these equilibrium points are quite interesting and important. So you want to actually understand what happens around these equilibrium points once you find them. And there are several types of equilibrium points that will tell you the behavior of your system. You have like nodes. This means it's a resting state. All your trajectories, depending on where, independent of where you start, you're going to end up here, and you're going to be at rest and do nothing then. Either you can have something that is unstable, so this will repel trajectories. You don't want to go there. A saddle is going to depend on the direction. So if you're coming from here, you're going to be attracted, but here after, you're going to be repelled. And then the oscillations, they're related to this kind of spiral states, either converging, so these are going to be damped oscillations, or diverging, so this is going to be amplified oscillations. And what we see with our system is that we have a very interesting bifurcation route. And so first, we start at high temperature with no oscillation, so we have a resting state. And then when you increase the temperature, you create spiral state. Sorry. That allows for oscillations. This is as when you decrease. And what is interesting is to study these bifurcations and to understand, so the bifurcations, they correspond to the creation, annihilation, or switch instability of equilibrium points. And we see that we start with a stable node, and at the end, we have an unstable spiral. And one of the questions is how mathematically similar to what happens in biological neurons is our system. And so for that, you first need to model biological neurons. So what most people do in biology is they take this, the system on the left, that is 4-dimensionally, is biologically accurate, but is very hard to deal with. So I used a simplified version that has only 2 dimensions. And this is because it's much, it's going to be simpler to compare to my system that has also 2 dimensions. And you will see that it also has quite complex behavior, even though it's simple. And what we demonstrated is that this is the bifurcation route for our DNA system. And so when it's hot, you have rest on a stable node. And then you have some bifurcations with creation and annihilation of these new equilibrium points. But what matters is at the end, you have an unstable spiral and oscillations around the limit cycle. So this is your oscillator. And if you look at the biological neuron, you have something very similar. First, when it's hot, rest on a stable state, so no oscillations. And when you decrease the temperature, you will have also an unstable state and trajectories that oscillate around this point. And this is not trivial, because if you look at the literature and find like other canonical bifurcations and try to compare how they would behave with a switch in temperature between on, off, you see that such system, either they would have problem to create a limit cycle, so to have nice oscillations. In this case, you have also an oscillation when the system is supposed to be off, or they have a difficulty to destroy the resting state. And when you're supposed to be off, you still have damped oscillations. And so it's not so trivial that we have a good response with our system similarly to this kind of biological neurons. And now that we have this understanding, we can use the system to encode thermal messages. So we first did in a digital way, which means we operated the chemical neuron as a digital communication channel. So to pass 10 bits thermal messages, what is a thermal messages is a sequence of 0 and 1s. So a 1, we want a spike, and how do we make a 1? We have a cold temperature, and when we have a cold temperature, the system is supposed to spike, and then when it's hot, we go above the bifurcation temperature, so the system is at rest, and you have no oscillation. And we did that on 38 inputs, the different sequences. With triplicates, and we got a 92% accuracy, so we're pretty happy. But then if you look at biology, that's not the way biology computes. biology encodes information mostly, for instance, with firing rates. So what we did is we encapsulated our programs into very small droplets, and you will get more details on the droplets in the next talk. And these droplets are roughly 50 micrometers droplets, and you can vary the composition of what you have, and then you can have these droplets, around 100,000 of these droplets, in a temperature gradient. So here is going to be cold, here is going to be hot, and you look at what happens, what are the dynamics in each of these droplets. And then you map the oscillations in a diagram where you varied the exonuclease, so the degradation, and the temperature. And we thought that we could vary the frequency by a factor of 3, which can help us to encode information. So in summary, we were able to design a DNA thermosensory neuron that shares similarities with biological neurons as far as the bifurcation routes are concerned, and we were able to encode digital and analog information. And to finish, I would like to acknowledge the help of the different PIs that followed me during this project. And special thanks to Anthony. So you're going to know more about his research in the next session. And yes, he was more than my PhD supervisor. We've been doing great science for, at least I enjoyed a lot in the last 9 years. But I'm happy to see a lot of the people that work with him that are still working on this project, and that we can continue working together. And if you're interested in this project, you have here the QR code and also the bioRxiv version. Thank you for your time.
Questions?
Thank you for the talk. What is the purpose of mimicking spikes? Do you want to interface then with real neurons?
In this project, no. So one of from the DNA data storage or computing, we would like to be able to process information that is not related to the sequence of the inputs, but for instance, the dynamics. If we imagine a database where we don't care so much about the sequence of certain queries, but the fact that certain queries are coming at a certain frequency, and so this kind of information processing that depends on the dynamics more than directly the content. And so, of course, this system cannot do that. It's a very simple system. It's the simplest that it's still interesting to study, because you only do such oscillatory dynamics with 3 strands. But of course, the next step is to study similar bifurcation or similar circuits, but more complex dynamics.
Okay, my second question is it by chance that you find the same dynamics as neurons? Because DNA is completely different biophysical process. So it's just by chance you find the same dynamics?
Not completely by chance, because here it's engineered. Like we wanted a system, we wanted to mimic a system that was at rest, at hot temperature and oscillating at warm temperature. And in fact, if you look more deeply at the equations, it's just statistical physics, because temperature, you will have like, balance between the entropy and the energy of binding. And it's the same exactly for the ionic channels. You will have a probability distribution of open and closed channels. So if you look at the physics, it's the same physics.
Hi, thank you for an exciting talk. I wonder, so neurons can fire from different regions of the cell. And you probably alluded to it, are you planning to make like different structures with your DNA neuronal models? And if so, like how are you, how do you see this scaling in terms of spiking and computation?
So very good point. So here we mimic thermosensory neurons because it was easy to give like a temperature signal, a temperature input to our system, because for instance the droplets, it's a closed system. But of course, to improve this system, it would be interesting to be able to get a stimuli, for instance, light, so so heat, light, or electrical input. So maybe some electrochemical reaction. So this would be interesting.
This work was really cool. I have two questions. How stable or how long can you run your measurements?
So the measurements, it's more like the system, so the measurement is done with fluorescence, and this is quite stable and we don't have problem. It's more the system, how long can it oscillate for. So when in the record was done for a former PhD student, should I go? Here maybe, sorry. And it's 1 week, and it's roughly 100, no, like back? That's okay. Hey, Macarena, sorry. No? Okay. Okay, sorry. So basically, it's 100 oscillations over a week. So if you, the thing is, if you want very non-damped oscillations, you need some optimization. So of course, one of the things you want to improve with the system is it's not very fast. You get one oscillation in roughly 20 minutes. But because we want something very simple, if you're interested in other type of dynamics and that don't need to make very like sinusoidal type beautiful, you can go faster and you can have the system works in the order of several days.
But it means that you add always new enzymes, or?
No, so the system is always producing new DNA via DNTPs, so this is the fuel. And the interesting point is that the concentration of the fuel doesn't affect the dynamics. So we can put fuel for 1 week for 10 times more. We can put 10 times more fuel than what we need, and so it doesn't affect the dynamics.
Yeah, the DNA, but what about the enzymes?
So the enzymes, at the temperature, what I told you is you can run it for roughly a few days without so much variation.
Okay, and how many data points do you need to make such a 2D diagram?
So I don't know if I needed them, but we can have between, like, we can have 100,000 data points in one experiment, and this is exactly what you will learn in the next talk.
Last question.
Just a quick, first, thank you very much for this exciting talk. It was really nice to see this. Also, if you would, for example, stop the system, could you restart it?
Yes.
Oh, okay.
So that's what we did with the 0-1. It's basically that. You run it for 2 weeks, 2 hours at a certain temperature, and then you increase the temperature, so and this restarts the system.
Oh, okay, so it's always put on standby for
yes.
the 0? Okay.
Exactly.
Thank you.
Thank you.
Thank you so much.